Hello everyone and welcome to today's live broadcast, Why Variants Matter, NGS Bioinformatics Simplified for Oncology and Virology, presented by Dr. Condie, Carmack, and Dr. Ahmed, Mamoud. I'm Susie Valdez of LabRoots and I will be your moderator for today's event. We're excited to bring you this educational web seminar presented by LabRoots, the leading scientific social networking website provider of virtual events and webinars advancing scientific collaboration and learning. Today's event is sponsored by Vela Diagnostics. Vela Diagnostics is a worldwide supplier of integrated life sciences and diagnostic solutions that help provide customers with valuable molecular information. From scientists striving to make research advances and technicians reporting the information necessary to identify, monitor, and treat diseases. Before we begin, I'd like to remind everyone that this presentation is interactive. We want to hear from you. Questions, comments, and even answers can be submitted via the green Q&A button at the lower left of your screen. We'll try to get to everyone, but if not, we will make sure to follow up with you by email. You can also enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you cannot hear or see the presentation properly, let us know by clicking on the support button at the top right or the Q&A button at the lower left. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, please click on the CE button located at the bottom left-hand corner of your web page and follow the process of obtaining your credits. I'd now like to introduce our two presenters today. First, Dr. Ahmed Mamoud, Director of Scientific Support for Vela Diagnostics. Dr. Mamoud is an experienced scientist with both hands-on lab and bioinformatics experience with degrees in molecular biology and bioinformatics. Dr. Mamoud is currently the Director of Scientific Support for Vela Diagnostics, as well as the Bioinformatic Application Scientist. Dr. Mamoud's team handles all the technical wet lab and informatic support for Vela Diagnostics qPCR and NGS workflows, assisting with pipeline creation for various NGS workflows and robotic application writings for automatic systems. He has experience developing the qPCR and the automated NGS workflows, as well as extensive experience in NGS data analysis using multiple sequencing platforms. He has held positions in various areas of biotech industry, with institutions such as GeneDx, Pittsburgh, and Columbia University Medical Center. Dr. Mamoud is also an avid iOS developer, creator of computer apps gearing towards the science sector. With him today is Dr. Carmack, our second speaker. Dr. Condie Carmack is the Vice President of Precision Oncology at Vela Diagnostics. Dr. Carmack received his PhD from the University of Utah in experimental pathology and did his postdoc in Philadelphia, Pennsylvania, and in San Diego, California, in mouse genetics and molecular biology. He is the author or co-author of 23 peer-reviewed journals. He wrote the first paper on quantitative PCR and the second paper on multiplex PCR. He served as a science advisory board of Life Technologies, Inc., and is an expert in clinical PCR. Dr. Carmack was the co-author on the first paper creating human monoclonal antibodies for the immunodeficient mice. Mice serve as a foundation for creation of the current immune checkpoint blockade antibodies. He also served as the general manager of the cancer genetic lab at Baylor College of Medicine before joining Vela Diagnostics. I will now turn the presentation over to these gentlemen. Dr. Carmack, Dr. Mamoud, welcome.
Thank you so much, Susie, for that introduction, and thank you, everybody, for coming in and listening to me and Condie's presentation. So let's get started here. Our title is Why Variants Matter, Bioinformatics Simplified for Oncology and Virology. So what we hope to get out of this, the outline will be, I'll give you a brief introduction to next-generation sequencing bioinformatics pipelines, how they apply to the field of virology, and then Dr. Carmack will then go over the oncology section. So key learning objectives here is to just understand the basics of bioinformatics as it applies to next-generation sequencing, and how we take all of this sequencing data and generate it into more meaningful information. Second thing we wish to achieve here is to have you understand how the solutions that we have created for virology and oncology help to make your workflow and your processes a lot more easier, a lot more automated. So let's start with the bioinformatics pipelines here. So there are a few components of a pipeline, and they're usually attributed to files. First file that you'll see is the FASTQ file. So the FASTQ file is very similar to its sister file, the FASTA file, which some of you may have heard of. So this is essentially what a FASTQ file will look like. You'll have your same information that your FASTA file will have. We'll have basically a sequence identifier and your sequence itself. In this case, for a FASTQ file, you'll also add one more component, and that's a quality score. So this isn't really interpretable for as humanly read in interpretation, but it is encoded in ASCII format for brevity. And it encodes something called a Phred score. So the Phred score is the probability that your base has been called in error. Phred scores range from 0 all the way up. You can actually have Phred scores higher than 41, but basically the higher your Phred score, the better your probability is that your base has not been called in error. So that is a FASTQ file format. That FASTQ file will go into an aligner. So we're here now. So the alignment phase uses that FASTQ file and a genomic reference to align all those reads onto the reference and assign it a score. So there are different types of alignments. There's global, local, and pairwise alignment. So global alignment is basically taking the whole read and trying to align that whole read to a reference. Local alignment takes parts of that read and tries to align it to the reference. And then pairwise will take two reads and try to align it to themselves, to each other. There are a variety of different open source software that can actually do this for you. Some of them are mentioned here. BWA is probably BWA and Bowtie are probably the more popular versions out there. What gets outputted by this process is called a SAM file. So the SAM file stands for Sequence Alignment Map File. And I'll show you a description of it here. So this is a SAM file here. And what the SAM file has is basically what the FASTQ file had, which is the sequence information as well as that quality score information. But what it does add is key information as to how that read was mapped. So I'd like to point, I'm not going to go over all of the elements here, but I go over some of them that I think are a little more relevant. This right here is a mapping quality score, so it's basically how well your read has mapped to the reference. Another key information element is the cigar string, which is right here. So this basically gives you an overview of how the read has aligned over the whole course of the read itself. So in this case here, this one's a simple one. We have 100 reads that have been mapped. If we look at this bottom one here, you can see that it's in a different annotation. You have 50 mapped reads, and then you have a 4,700 base pair span. N would be indicating an intronic region, and then 50 base pairs have been mapped. So in this one, you can decipher that the beginning of this read was mapped to one part of an exon. Then there's a split, and then there's another 50 reads that have been mapped to the other portion of the exon itself. So a lot more information is being added into the SAM format for the subsequent processes of variant calling. So that is the next process involved. That is variant calling. So it uses that, first off, actually, the SAM file is converted into something called a BAM file. So that BAM file is an identical copy to the SAM file. It has just been encoded in binary format and indexed to make it accessible to the computer. So we take that BAM file over and we run it through a variant caller. The variant caller will just call mismatches between the reference and the actual read itself. There are a variety of different variant callers. As you can see here, some of them are open source, some of them are not. But all of them output a file called the variant caller format. So the variant caller format looks like this here. So you'll have a variety of different headers which will give you descriptions of some of the tags that are included in the VCF itself. So usually what you'll have is some of the common tags will be chromosome, ID, position, your any ID for the actual SNP itself. So if you have any RS numbers or anything like that, identifiers will be placed here. Then you'll have your reference sequence, your reference read, your reference base, sorry, and then your alternate base right here. And then you'll have your quality score and any filters that have been placed in as a metric. So if we take, for example, let's take, for example, this specific SNP here. So in this specific SNP, there is a reference allele of an A, and then two different reference alleles that are attached to this RS number, and that's a G and a T. The quality here is 67, which is good, and that the filter indicates that it's passed. So if you take a look at the one above it, the filter says Q10. This is because in this specific, with this specific variant, we were only able to get a quality score of 3. So in this filter parameter, what they've specified is anything less than Q10 gets flagged here. So let's go back to the one that we were looking at here, and you'll get some of the tags here. So here is the number of specific samples with actual data. So samples that actually have any of these SNPs would be 2 samples here. Your depth and your allelic frequency, among other tags that you can decipher from the headers up here. Additionally, you'll also have some information related to each sample. So if you look here, we have our genotype. So this genotype basically tells you for this specific sample, we have a reference, and we also have called both of these alternate alleles here. So we have 2 different alternate alleles that have been called here. We also have the genotyping quality, and we have the depth of sequencing. So a lot of information that helps you get as much information as possible about the variants you're calling and allow you to infer on if they are actual real variants or sequencing artifacts or anything like that. So I'd like to go into the genotyping and variant calling for our HIV analysis workflow. So this is just a brief diagram of the processes. So we start with a FASTQ file here, and we go into sequence QC, where we generate some of the information as far as run throughput and our base quality. So that's off of the FASTQ file. Then we do mapping, so we align to a reference, and then we do some read filtering. Then we take all those reads and we assemble a contig. So a contig is a consensus sequence of all the reads that have been generated. We then do a further alignment and generate a BAM file. That BAM file goes into the variant calling software. It generates that VCF, and we use that VCF to parse data and actually get information from the HIV database. We also do the genotyping as well based off of the contig that we generated. So we'll go through all of this in the next group of slides. So this is the sequence QC. These are screenshots from our actual reporter software. What you'll see is you'll get all your sample information, and you'll get the total bases, total reads. You'll get the quality score for each base, for each base of the read here, and it'll come up in quantile charts over here. You'll also get information as to how well the quality has been, how well the QC, the quality scores have been for the actual read itself, right here, and for the cumulative run as a whole. You also get read length distributions over here as well. Here we start mapping those reads to a reference sequence. So once we align all these reads to the reference, we do filtering. So we do several rounds of filtering. So our first round of filtering actually is to filter out any reads that weren't mapped. So unmapped reads are reads that haven't been mapped to any of the reference sequences that we have in our database. We then take a look at the mapped reads here. In mapped reads, we do a subsequent filtering based off of where they've been aligned to. So we have non-specific reads here. So these are reads that have not aligned to any of the regions that we are looking for. So these regions can be, we can look at it as specific reads here. So non-specific reads are reads that haven't aligned to either the control, protease and RT, and integrase regions. Discarded reads are reads that have a length of less than 25 base pairs long, or less than 25% of the read has aligned to any of the regions. Now we're going to do assembly. Assembly is basically the process of generating that consensus sequence and creating that contig that we're going to use for genotyping. Before we do that, we need to downsample. So our sequencing usually typically produces about 5,000 to 15,000 X coverage. If we were to use all of those reads for the assembler, it would provide for a very slow process and actually introduce errors. So what we do is we downsample to about 1,100 X coverage for each region that we're doing. Only the downsampled reads are assembled into this contig, and we generate two contigs, one for the protease and RT, and one for the integrase itself. We filter out for short and low coverage contigs, so those with a median coverage of 250 base pairs or less are filtered out, and contigs that are greater than 98% are merged. So this is variant calling alignment here. So what we essentially want to do is we want to take the contigs that we've generated and try to align them to the reference genome. So when we genotype, when we go for genotyping, we're actually going to align the contigs to over 4,000 genotype references that are in a database. We'll talk about that when we get into genotyping, but what I wanted to get into right now is actual variant calling. So if any of you are familiar with oncology, what you normally do when you're running variant calling is you're going to take a reference sequence and you're going to align the reads to that reference sequence. Very simple process, relatively simple process. Now, if you look here, variant calling for viral genomes is a little more complicated than that because there is greater variation from strain to strain. So if you notice this example right here, you see that these reads don't quite align to this reference sequence at the moment. So what do? Well, we have another component that we generated before, and that was for genotyping, which is the actual contig itself. So our approach is to actually take the contig that has already been aligned to these reads, use that contig as a genomic locator to the reference. Once we've found the location that this contig is placed, the only possible place that those reads could align to is within that contiguous sequence. So we use that contig as a locator. We call variants directly off the reads after that point. So we basically recreate, we re-coordinate the reads based off of the contig itself. So here's just a visualization plot off of IGV that that just shows our approach versus the conventional approach. So if you look at the top here, you'll see all of these different colors just encode for just different types of variant calls here. So you can see there's some spots here that are open, basically indicates that no reads will align to these regions. But when we use the exact same, when we use our approach on the exact same data, you'll actually see that we have much higher coverage and much better variant calling with our approach. So once we've called the variants, we output them on our reporter interface here, and what you'll get is a very simple, streamlined table with the position, your gene, your amino acid mutation, variant frequency, your coverage, reference coverage, variant coverage, your p-values, and also if you click this link, you'll get a visual look at your read depth. Then we're going to take a look at genotyping here. So what we do is we generate a phylogenetic tree. This is pre-built, and the database consists of over 4,000 protease, reverse transcriptase, and integrase regions. And what we do is we take that contig, and we actually blast that contig over that database. So we try to find the closest hit out of that database to our contiguous sequence. So by looking at the lowest common ancestor here, we can determine what type of strain that the actual viral, that the sample is. So this is just a little overview of how the phylogenetic tree is set up here. And essentially anything under the lowest common ancestor is the same genotype, so we'll call that. So after that, we have our reports. So we have 3 reports. The first report is a QC report. So the QC report will go over all the sequencing information, all the metrics that need to pass in order to generate a sufficient NGS run. So we have run quality controls and sample quality controls. And within this, we have different controls that are for separate parts of our workflow. So we have a control that will only apply to our extraction. We have a control that will only apply to sequencing, amplification and sequencing, and then we have controls that will control for the whole process as well as variant calling as well. So you'll have all the information you need to confidently provide a report and know that your sequencing has been done and performed correctly. The next report that we use is the variant calling report and the genotyping report. So this is essentially just another snapshot from the page that I have showed previously, but with genotyping information now. So here you'll see that we've generated 2 contigs of the subtype B. We have an integrase and a protease. And this is the median coverage for both of those contigs. We also have the target variants list, which will give you all the information you need for all the variants that have been called. The last report is the drug resistance mutation report. So this report is actually generated from the Stanford HIV database or the 2 other databases that we have in this section here. So essentially what we do is we take the VCF that has been generated and we send that to either a local instance of HIVDB or an online instance of HIV-DB, depending on the customer's preferences. From there, this automated report gets generated based off of all the VCF information that was submitted. So you'll get a report that has information for the integrase region, the protease region, and the reverse transcriptase region here. And it'll give you susceptibility information from Stanford, ANRS, and the REGA databases. Also with some comments about how these variants confer resistance to these specific drugs. So just to recap here, NGS is a fast and accurate way to get genotyping and drug resistance mutation information. That coupled with the Stanford HIV database is a very concise resource that allows you to get as much information as possible and as much streamlined information as possible for you to generate a report that has drug resistance information in a concise and streamlined report for you. With that, I'd like to send it over to Condie.
Well, thank you, Ahmed. I appreciate that. So today I'm going to continue the theme of variant calling and cover a couple of different topics regarding cancer variants. First, we want to define the different types of the cancer variants seen in cancer. Second, we want to talk about the origins of these variants. How did they arise? Also, I'm going to be talking extensively about databases that we use to collect and annotate these variants. Also, how do you know the difference between a true variant as opposed to an artifact of the processing? Sometimes this can be difficult. Also, once you get a true list of variants, how do you classify them? How do you report them? And to whom are you going to write the report? We'll also cover how the annotation is done, and is the annotation useful, predictive, prognostic, and diagnostic. Off in the future, someday, we'll be able to fix some of these problem variants in patients and probands using a number of biotechnology methods. So NGS, or Next-Gen Sequencing, has truly revolutionized the pathology analysis of tumors. NGS sequencing of tumors is the first time that we've been able to look at the nucleotide level of cancers. From the variants, we can associate genes, and from the genes, we can associate pathways to start to understand the complex nature of and the molecular biology of the tumors. DNA sequencing analysis is not always used as standalone diagnostics. It's actually part of a molecular toolbox that all of the labs need to develop. As NGS DNA sequencing of multi-gene panels becomes routine and standard for cancer samples, there must be an efficient and routine method for processing samples and for classifying variants. Determining which variants are biologically significant for cancer maintenance and development and which are not is not always easy, not even for the experts. Fortunately, unfortunately, I should say, there is no agreed upon standards as to how to do this just yet. Much of the work usually falls onto the pathologist, who must advise the oncologist in recommending the test order, the amounts of tissue needed, and the budgets involved. And will the tests actually be relevant and useful to the treatment of the patient? So there are a number of different choices that the pathologist and the oncologist must make, and the first decision is the type of NGS tumor to choose. There are generally three main categories of NGS testing. They're shown on the slide: gene panels, whole exome, and whole genome. Genome panels, these include tens to thousands of genes, are the most popular currently because they are designed to cover only the genes of interest that relate to the cancer genes. They may actually be part of a pan-cancer, in other words, covering all cancers. or maybe a subset, maybe solid tumor cancers. Or they may be very specific to an individual tissue. Gene panels are the cheapest to run, and they have the highest depth of coverage. They're also the easiest to interpret. Whole exome testing is done at several academic institutions, but is still experimental. The technical sequencing is very good, but we're just not sure of the value added by covering all of the genes at the nucleotide level. The question is it worth the cost? Whole genome testing is being done at a few places, but again, like whole exome, the value is questionable compared to the results. As the cost of sequencing and analysis drop, and as the databases get better, whole exome and whole genome will certainly be considered tests of the future. For routine testing, gene panels will dominate the market for the time to come. And even if the gene panels turn up negative, there's always enough time, there usually is enough tissue to actually generate another larger panel. Remember, you only need about 10 nanograms of FFPE DNA to run a gene panel. So now I'd like to just go through a couple of quick definitions. Genetic variant, these are alterations or genetic differences in a patient compared to the most common or reference DNA sequence. These can describe changes that are benign, pathogenic, or of unknown significance, something called VUSs, or variants of unknown significance. Variants can be of two different types. They can be normal, non-pathogenic genetic polymorphisms, or they can be pathological or pathogenic. These changes or mutations that usually cause frank disease, and in our case, cancer. These pathogenic variants can be divided up into a number of different classes. Single nucleotide polymorphisms, or SNPs, make up about 90% of the variants seen in the genome in cancer. Intragenic insertions and deletions, sometimes called indels, are often seen. These can be small to large. Also, structural variants, SVs, dot the landscape. These can be divided into two different types, copy number changes, as well as chromosomal fusions or translocations. Regulatory agencies have taken an interest in this area. The New York State has published NGS guidelines and goes through the minimal requirements for developing lab tests of different types of variants. This usually is about 10 positive controls or 10 retrospective controls of a particular type of variant. The FDA have also held workshops in the past few years about NGS and how to define the variants. They're very concerned that the variants will be used as companion diagnostics that are used for decision making to treat cancers with specific drugs. So variant analysis really begins with the tissue and how it's handled and processed in the operating room and in the pathology lab. FFPE, or formalin-fixed paraffin embedded tissue, is the most common way tissue is processed. When working with DNA and RNA samples, it's important to use neutral pH buffers and avoid aging the blocks more than necessary, as this will affect the results. FFPE can cause a number of chemical changes in DNA and also in RNA, including deamination, oxidation, cyclic base derivative formation, and methylene cross-link formation. In FFPE tissue, nucleotide transitions can occur. So the first step in ensuring robust variant calling in cancer relies on accurate base calling and alignment to the reference assembly. As Ahmed described, raw sequence data from high-throughput sequencers are moved through an analysis pipeline to call the bases, align them to the reference sequence, filter on quality, and finally call the variants. Vela provides an end-to-end instrument and bioinformatics pipeline, taking all of the different genetic variants into account. Now the second step, once you have a list of variant calls, is to classify, prioritize, and annotate them. In cancer, many of the labs are sequencing both somatic cancer tissue as well as individual as well as an individual's germline or non-cancer genome. This will give you a better result as to the true somatic variants, as you will be able to subtract out your inherited or germline variants. The tumor-matched normal is always the best control to determine which variants are germline and which are somatically derived. The problem is that the sequencing the germline is a quality check and not always reimbursed. So you'll have twice the cost, and the value of doing this control is still being debated. If this were a tumor-only bioinformatics pipeline, then we would filter the variant list against a list of normal or naturally occurring variants in the non-pathogenic population. This is the germline filtering to remove the non-pathogenic variants, and in this way we'll get a better list to curate of pathogenic variants. If this were a reoccurring tumor or a metastatic cancer, then it might be possible to go back and look at the previous variants that were sequenced in the primary tumor. And this would be important to understand why the tumor progressed and if the tumor has developed resistance mutations that would affect the current treatment. Again, this is an area that continues to evolve. So to filter the germline variants, we take the entire variant list and we match it to a database or databases of normal variants, which tallies the normal non-pathogenic variants that are normally seen in the general population. Here is a list of projects and databases that have been used to filter germline variants and decide which of the variants actually fall into the germline or into the somatic class. Remember that we, as humans, are all about 99.5% identical to one another at the nucleotide level, but there are still about 10 to 30 million variants that we differ at due to normal genetic variation. So there are a number of professional societies and organizations that have drawn up guidelines and recommendations as to how to handle genetic variants. You see on the slide there's the ACMG, the American College of Medical Genetics and Genomics, the IARC, the International Agency for Research on Cancer, the AMP, Association for Molecular Pathology, CAP, the College of American Pathologists, and ASCO, the American Society of Clinical Oncology. These groups are working together to make recommendations as to the best practices in the industry and for healthcare. So the AMP and the ACMG have gotten together and published joint standards and guidelines for the interpretation of sequence variants for germline and inherited disease. This was actually the first of a number of standards and guidelines paper. This was published in the Genetics in Medicine journal and occurred early in 2015. Subsequent to that guideline publication, the ACMG went on to publish a list of incidental findings or secondary findings that should be considered separately for clinical exome and genome sequencing, as patients may not want to know their genetic status as to certain genes for personal reasons. This is the most recent update, which appeared in the March 2017 issue of Journal of Molecular Diagnostics. Once we have filtered the germline variants from the somatic variants, we now have a list that should be considered cancer-specific somatic variants. This list is usually long, and most of the variants are passenger mutations. In other words, they're along for the ride and don't contribute to any pathology. As such, these variants are not druggable nor actionable, in that they contribute nothing to the overall survival of the tumor. The TCGA, the Cancer Genome Atlas, is a project from the NCI-NHGRI that has completed its work but is still releasing data in the form of papers and database submissions. CIVIC, which stands for Clinical Interpretation of Variants in Cancer, is an expert crowdsourced knowledge base from Washington University describing therapeutic, prognostic, diagnostic, and predisposing relevance of inherited and somatic variants of all types. There are now databases that coordinate the variant gene pathway information and now relate it to relevant drugs. The Drug-Gene Interaction Database at Washington University and the Pharmacogenomics Knowledge Base, PharmGKB, at Stanford University are two such databases. Currently, there are 402 potentially druggable genes listed in the DGI database. One result that came out of the TCGA sequencing project was that different cancers have different mutation rates. This analysis from the Broad shows a significant difference between melanoma and lung cancers, shown on the right-hand side, compared to inherited disease, shown on the left-hand side of the graph. This observation could have implications when treating patients with the new immunocheckpoint inhibitors. With regards to cancer testing, several professional organizations have established standards and guidelines to follow. In December of 2016, CAP, AMP, and ASCO issued joint guidelines for the standardization, interpretation, and reporting of sequence variants in cancer. This was published in the Journal of Molecular Diagnostics. This report, authored by Marilyn Lee, makes recommendations to pathologists and oncologists that assess the status of NGS-based cancer tests and establishes standard classifications, annotations, interpretations, and reporting conventions for somatic sequence variants. These guidelines prioritize the variants into actionable variants that are classified by being therapeutic, prognostic, or diagnostic. Also recently published by AMP and CAP was the best practice guidelines for next-generation sequencing-based oncology panel validation. This was also published in the Journal of Molecular Diagnostics. The ASCO AMP-CAP joint guideline system contained 4 tiers to categorize somatic mutation variants by clinical significance. These are based on diagnostic, prognostic, and/or therapeutic value. Tier 1 are variants with strong clinical significance. Tier 2 are variants with potential clinical significance. Tier 3 are variants with unknown clinical significance. Tier 4 are variants deemed benign or likely benign. You can actually see that Tier 1 and Tier 2 are broken down into 2 more levels, A and B, for greater resolution. Lots of things are changing in the sequencing world, and variant calling is no different. There is a lot of new information about mutated genes, pathways, variants, and the drugs to treat them with. There's also valuable information with regards to dosing routes of administration and outcome. No one can be expected to know or to recall all of this information. There is the promise of artificial intelligence, AI, and machine learning coming along to be able to help to make sure that the variant calls and the drugs assigned are the best possible choices for each patient. iCages is a bioinformatics tool from Columbia University that prioritizes and classifies cancer driver genes for a more personalized result. IBM Watson is a similar tool to help clinicians understand all of the data involved in clinical sequencing and precision oncology. Obviously, there's a lot of work that is involved in taking the raw sequencing data, filtering it down into the correct variants, and annotating them properly. Vela now offers an informatics surface that takes the raw data files and maps the sequences. It filters all of the bad reads out and then compares the variants to 46 gene and sequence databases that are available. These represent all of the somatic mutations found in 822 cancer-associated genes. It also pulls the information from over 200,000 clinical trials that are going on in the world today to do clinical trial matching. Vela then takes this data and summarizes it into an easy-to-read, easy-to-understand clinical report called therapies. This tells the doctor what he needs to know, what is actionable and what is druggable. In the near future, researchers are working on ways to detect tumor variants from normal blood draws. This type of testing will identify somatic variants for screening and for residual disease, and will give the oncologist an early heads up on cancer screening. A number of companies, including Vela, have cell-free DNA kits and extraction methods to isolate and sequence cell-free DNA. And finally, in the not-so-near future, scientists are working on a number of DNA manipulations and cellular targeting methods that will potentially modify the DNA of the patient and treat the patient at the germline level. Zinc fingers, talons, and more recently, Cas-CRISPR technologies could all be used for this purpose. So to summarize, there are new joint guidelines and standards that have been recently published by professional organizations. These guidelines recommend tier-based somatic variant categorization and reporting. Informatics can and should be automated. Variant prioritization can assist in the interpretation and reporting, but a final manual curation should still be done. Development of variant pipeline analysis requires careful review of variants and an understanding of the limitations of the current sequence databases. And finally, Vela has developed both hardware solutions and software solutions to solve NGS cancer testing problems. And with that, I'd like to turn it back to Susie.
Thank you so much for that wonderful presentation, Dr. Carmack and Dr. Mamoud. We want to get right to the questions. So here's a reminder to the audience members how they can communicate with us. Questions can be submitted via the Q&A button at the lower left of your screen. If you are unable to get to your questions due to time constraints, Dr. Carmack and Dr. Mamoud will be answering submitted questions via email. So let's begin with our first question. This question is for you, Dr. Mamoud. How important is it to analyze the integrase gene for HIV DRMs?
I think it's very important. It's just as important as analyzing the protease or reverse transcriptase regions, specifically because there are integrase inhibitor drugs that are being developed to target this specific region. So things like elvitegravir and raltegravir specifically these drugs are specifically targeting the integrase gene itself. So in order for you to properly diagnose in a scenario where you are going to prescribe this specific drug to a patient, you would essentially need to be able to know whether they have any mutations or DRMs in that viral strain that would pose some resistance to that drug.
Thank you so much for that, Dr. Mamoud. We actually have another question for you from an audience member. How is bioinformatics development different for use in the analysis of viral RNA versus, let's say, human DNA?
Okay, so I think we touched on it a little bit with the presentation. The viral genome is much less complicated to some degree versus the actual human genome itself. But what's less complicated about it complicates it even more, if that makes any sense. The genetic variation from strain to strain makes it much more difficult to actually sequence and perform an alignment and variant calling on it. So usually what you end up having to do is you cannot do any, as far as targeted primer sequencing, you can't really do that with a viral genome because of the variation. You'll have, you will tend to have SNPs under primers, and if you have primers that are amplifying all these specific regions that you're targeting or the hotspots that you're targeting, you could be targeting, let's say for HIV, maybe over 300 variants, and those variants will, you might have 80, 90 primers or something like that. You're bound to have some SNPs under those primers that might drop out those sequences themselves. So what we end up having to apply is sort of a whole genome shotgun sequencing approach to a relatively small genome. So you might have something that along the lines of regions that might be about 1,000 base pairs long, but you have to apply this whole genome approach to those to the viral genome in order for you to actually be able to get robust and efficient sequencing data out of it.
Thank you, sir, for those answers. This next question is for your colleague, Dr. Carmack. How do curators weigh the various pieces of information found in literature for variants and make a determination of the importance of that mutation?
That's actually a very good question. Probably the best way to answer that is with experience and also curating, going through all of the databases and understanding, what the guidelines of the professional organizations are. But it really just takes experience and looking at outcome data that accumulates over time. So this is, again, one of those areas that continues to progress.
Thank you. We have one more for you, Dr. Carmack. Are there limitations to the cell-free DNA kits currently on the market for variant identifications, and will your company be putting out a product in this arena?
We will be putting out a product in this area. Again, this is one of those research areas that continues to expand. Some of the technical problems with cell-free DNA, of course, is getting the background down so that it is economically feasible to be able to sequence just the cell-free or the DNA from the tumor. So identification of the tumor from normally released cell-free DNA is still an issue. There are some technologies, methods to actually lower the background or to actually amplify the biomarkers that are needed. It always helps if you know the biomarkers that you're looking for. In other words, if the person has a leukemia or a biomarker that is a signature for the disease, to be able to look for one or two variants rather than a whole slew of variants.
Thank you, gentlemen, for those wonderful answers to those questions, and thank you so much for your presentation. I'd also like to thank LabRoots today for making today's educational webcast possible. Today's webcast will be available for on demand for up to 6 months. Keep an eye out for an email from LabRoots alerting you with this webcast and how it will be able to be replayed. We encourage you to forward that announcement to your colleagues who may have missed today's live event. Thank you so much for joining us again. We hope to see you next time. Goodbye.